Hi, everyone. This is Tara Coleman, the Executive Director of Children's Leukemia Research Association. In celebration of our upcoming 60th anniversary, we're checking in with our current and prior grant recipients. And I am very pleased to welcome Dr. Zakrzewski from the Center for Discovery and Innovation in Hackensack, New Jersey. Dr. Zakrzewski is a pediatric hematologist oncologist with more than a decade experience in hematopoietic stem cell transplantation and cellular therapy. He is the head of a laboratory focused on immuno-oncology and is one of the founding principal investigators of the Center for Discovery and Innovation and an attending physician at Hackensack University Medical Center, where he treats patients with a wide range of malignant and non-malignant diseases, including primary immunodeficiencies. He is a two-time grant recipient from CLRA, and we're here today to talk about his currently funded project for this year. And that is, I'm going to let you explain it. So can you tell us a little bit about this year's project?
Yeah, sure. No, thank you so much. Yeah, so we are working with a biotech company. They are basically in the same building. Just moved in earlier this year, but I started working with them since 2022. This company is developing platforms for immunotherapy for a wide range of cancers. So they came to me. They just started out in like 2020 during the pandemic, and then needed collaborators to really get things up and running. And so I knew them from previously. We had a relationship, so I developed a plan with them and they suggested pediatric AML as a target for just the initial development and clinical translation of the platform. And we started generating preliminary data. I funded it with basically just other grants, right? It was just to get some proof of concept. And so with that in mind, we showed that the targets are expressed, very unique targets, non-protein targets, and very specific to cancer and not found on any healthy cells. But we found it on AML, on all of the cell lines we tested. We went to patient cells as well, and we found it also on patient cells. And so then we knew with that information, right, that is, now we had something that we could use for grant applications as well, especially for seed funding, right? Not for the, of course, for a big grant, you need more, you need also some in vivo data. We didn't have that yet. But we went for the Children's Leukemia Research Association at that point because the technology is very innovative. A company is already involved, which is great to accelerate, clinical translation along the way. And we had in vitro, very, very convincing in vitro data suggesting that this should be safe and that this would likely be effective. And we did also do some killing assays. So we had some in vitro based, efficacy data as well at that point. So with that, I applied, and we got the grant. And with that grant, we were able to really create a very robust data set and then apply for bigger grants. So I was just awarded from Alex Lemonade Stand Foundation a $250,000 grant for this project. So that grant will become effective January 1st when the Children's Leukemia Research, Association money will run out. Basically, so it will be, we already have it lined up now, the bigger one that will fund us for the next 2 years, and a significant amount of money. So that's great. In addition, we submitted an ASH abstract in August, and it will be presented. So this study funded by CLRA will be presented as an oral presentation at ASH in a few weeks.
Oh, that's fabulous.
Yeah, so that's really, these are great accomplishments. We would not have been able to do this with this small grant that we got early this year. And yeah, and now we are basically in good shape. We are basically now safe for the next few years for sure. The company is raising money. We anticipate we are already discussing with my colleagues at the Children's Cancer Institute in Hackensack the clinical component of this project. So we are highly confident that within the next 2 to 3 years we will start treating patients, pediatric and maybe also adult, but we'll definitely do pediatric upfront, and we will not wait for adult AML first to be completed. We'll do the pediatric arm of the trial.
Right.
in parallel.
That's fantastic. That's such great news. Congratulations. It's a testament to your hard work. I know that when we met with you, when we had the pleasure of visiting, you were already making great project progress. So that's very exciting. And we're so thrilled again to have you as part of our little family. Any challenges with this year's project in the research, or has it been smooth sailing?
Well, it's we are really, you know, fortunate that the company is right there. So we, you know, I meet with them all the time. They're small, but it's been great for us to basically to share our data with them, and they are adding from their end a lot of the toxicology and pharmacokinetics, all these things. We don't have to worry about that at all. And then we apply to grants for grants together. I get the letters from them. So it actually has been a very, well, I would say, right, we are well supported, and we are really in a great position here to move forward quickly. We have a clinical fellow working on this, so I was able to find, you know, a clinical fellow who's doing this now for 2 years. She's with the lab at least for another year, and we just applied for a fellowship grant for her as well for this
That's
exact
great.
project. So we'll find out. That's the St. Baldrick's Foundation Fellowship. So we'll see in a few months she gets the grant, and if she gets it, she has another 2 years that she could stay. So
Oh, that's great.
we have a lot of things going on right now. This project we started, as I said, like 2022 with no funding, and just, you know, talking, and then you know, doing some testing on cell lines, and now it's huge. So it's been fantastic.
Yeah, that sounds fantastic. So we're hearing, obviously you talked a lot about AML. We talk to patients with AML every day. And what are there any particular challenges or obstacles that are unique to a pediatric AML patient versus an adult, or are you seeing the same
Well,
trends?
there are issues. Certainly, there's some significant issues when it comes to immunotherapy of AML, because AML is one of those diseases where it's been very challenging.
Yeah,
So the targets is not the only issue. Everyone's looking for the best targets, right? Even if you had the greatest targets, right, that are 100% specific to the cancer cells, and that are definitely not found on any healthy tissue, any healthy cell, even if you had that, and we think that's what we actually do now. But even then, it might not be easy to get great clinical results, because you're still dealing with very dysfunctional T cells in these patients.
yeah,
So we're injecting the molecules that these are basically T cell engagers. So they have to redirect the patient's own T cells to, you know, to the cancer, to the bone marrow where the cancer resides, and then kill the cancer cells in the bone marrow. But if the T cells are dysfunctional, even if they're engaged properly and redirected appropriately, they might still not kill as efficiently as they should, because of that immunosuppressive microenvironment in the bone marrow. And AML is well known for that, immunosuppressive microenvironment, and T cell dysfunction is a big issue in AML. So that remains a challenge. We will have to design smart clinical protocols to deal with that. And so we're discussing already how we would design these trials, but most likely it will be needed to at least get some chemotherapy upfront and reduce the disease burden. And ideally, you would come in with your immunotherapy after you use at least a partial remission, so the patient's T cells have recovered a little bit, and T cell immunity is not as compromised as it usually is in the beginning when you're newly diagnosed. So so the timing of immunotherapy is important. So we'll keep that in mind when we design the trial. But that's a issue with the CAR T cell therapies and any T cell engager therapies for AML. So that's not unique to us, but it's been a challenge for the field for years now, and that really in a big contrast to, let's say, ALL, where or other B cell malignancies, lymphomas, leukemias, even multiple myeloma. We have great success with CAR T cells, for different targets, not just CD19, and also T cell engager therapy. So that's very different. But AML is just really, really tough. So we will see. We are ready for this. We are not going to shy away from this, because it's a bit risky. It's a risky to treat with immunotherapy, but we haven't really moved the needle for AML in decades. So this is really the time to really, to go all in. And so we are doing that.
That's great. Yeah. So like obviously I'm not a research doctor such as yourself, but everything that you're saying is everything that we are hearing as well, and from other grant recipients and from just the industry, that AML has been the toughest one for years, for
Right.
years.
And not in all fairness, that applies to adult AML patients as well. And
Yeah.
mortality in adult patient is terrible, right? In patients in their 60s, 5-year survival is basically less than 10%.
Wow.
So that's really terrible. So this will benefit all age groups, what we are doing, not just pediatrics, but
Great.
we have a strong focus on pediatric AML. And as I said, we are not going to go for adult AML first and pediatric second. We'll do both in parallel.
Both in parallel. That's great news. That is great news. I'm looking at my next couple of questions, and you've answered a lot of them already. And are there any new, other than the ones that we talked about, any new collaborations or technologies in the leukemia research field that you think will be especially impactful?
Well, yeah, there is a lot going on in terms of cellular therapy beyond CAR T cells, and at this point, right? There's
Yeah.
interest in using, for example, natural killer cells as an off-the-shelf product. There's cell lines that can be used, but there's cord blood as a cell source. Even pluripotent stem cell derived NK cells are now different. You know, they're different centers who've brought them to the clinic already. So chimeric antigen receptor engineered cells, different types of effector cells beyond just the T cells, that have been now successful for more than a decade. But it's NK cells are up and coming. So there's a lot of interest in NK cell based therapies, including also NK cell engagers. So not just T cell engager therapy. So there's a lot going on with NK. So we are getting into the NK cell field as well. And that includes the same technology. So we are already, we designed the CAR constructs already, and our plan is to make CAR NK cells for this. And with the same targets we are targeting with these, molecules. T cell engager molecules, but now we'll be doing it with CAR engineered cells. CAR T and CAR NK is the primary focus. We might even look beyond NK cells. Macrophages is another potential source. And so we are looking, at a wide range of effector cells that could be used as an off-the-shelf product. So you don't have to manufacture, for the patient using their own cells, and then it always takes some time. You have to harvest the cells, send them to the company, or you do it in-house. But it takes usually a significant amount of time before you actually have the cells, versus off the shelf where you just have them and you can just thaw them and infuse them right away. So NK cells are off the shelf, so that's a big logistical advantage. Also, it's more cost effective.
It's amazing to think about that, and the fact that we're coming up on 60 years, and 60 years ago, I don't know any of the people that were here or around, right? But the advancements and how important this work is, your work, our work, 60 years ago, we would not have had the same conversation. So it's very exciting to be a part of it, and very exciting to have, you with us doing the great work. So this we're going to wrap it up with this one. But what impact on the future of blood cancer have the grants you have received from CLRA have?
Well, the impact is more like a long game, right? These things take time, right? So these grants really, as I explained it, right, they help you to get more preliminary data, because these are small grants, right? From CLRA. So you have to be smart with this money, but then you get the bigger grants, and then you have another, that's exactly what I got now here for this current project. So it gives me another 2 years, and we are applying for more grants, right? So but that's basically already the minimum amount of time we need to also develop the investigational new drug, because that money, I don't need to raise money for that. That's the company's job. But that's millions of dollars, right? So that's a bit beyond what I can get. But that's all happening. So the impact will be the clinical trial. Patients will receive these treatments, and that will happen within the next 2 to 3 years.
Wonderful.
Yeah.
Such great news. It is such great news. Well, I want to thank you so much for your time, and I am going to let you get back to the lab, so to speak. And please tell everyone that that we have met there, that we send our best wishes to all of you. And I've learned a lot, and I'm so grateful to have met you and for you to take the time out of your day to spend with us and help us celebrate 60 years.
That's exciting. Thank you so much.